Hello. In this video, we will talk about Salmonella. Salmonella belongs to a single genus, Salmonella, containing one dominant species, Enterica, which is itself divided into subspecies. Almost all strains responsible for human infections belong to a subspecies also called Enterica. Salmonella bacteria have a specific nomenclature with more than 2,500 serovars, including Salmonella typhi, Salmonella enteritidis, and Salmonella typhimurium, which are responsible for various forms of infection. Salmonella can be responsible for two clinical presentations. On the one hand, major serotypes, such as Salmonella typhi and Salmonella paratyphi, cause typhoid and paratyphoid fever, a systemic febrile infection that can be severe. On the other hand, minor serotypes, which include other Salmonella species, mainly cause gastroenteritis, known as minor salmonellosis, which is more common but generally less severe. Typhoid fever is a prolonged septicemia of digestive origin caused by Salmonella typhi or paratyphi serotypes A, B, and C. It is a disease that affects only humans. The clinical picture is characteristic. Incubation is long, lasting 7 to 10 days, with high fever reaching 39 to 40 degrees, and a dissociation between pulse and temperature is often observed. Typhoid fever also presents pathognomonic symptoms that can guide diagnosis. Pink lenticular spots may be observed on the base of the chest, along with Dugace angina, corresponding to ulcerations of the tonsils. In terms of the epidemiology of Salmonella, typhoid fever is common in developing countries, particularly in Africa and Asia, with a high incidence in areas with poor hygiene. The reservoir is strictly human, and transmission can occur directly or indirectly, through contaminated water or food. The infectious dose is relatively high, in the range of 10 to the power of 5 to 10 to the power of 6 bacteria. Non-typhoid salmonellosis mainly manifests as acute gastroenteritis. The reservoir is animals, especially poultry, and the incubation period is short, lasting 12 to 36 hours. Typical symptoms include nausea, vomiting, diarrhea, high fever, and hyperleukocytosis. It is often the cause of food poisoning and resolves spontaneously without treatment in most cases. As for other Salmonella, they are present in both industrialized and developing countries. Their incidence is higher during the warmer months. The main reservoirs are animals, particularly poultry and cattle. The pathophysiology of Salmonella infection begins with the ingestion of bacteria, which cross the intestinal epithelium via M cells. After phagocytosis by macrophages, they spread to the mesenteric lymph nodes and can then enter the bloodstream, causing septicemia. The diagnosis of typhoid fever is based on the isolation of Salmonella typhi. In the septicemic phase, blood culture is the gold standard test, with stool culture being useful at a later stage. Serology is an indirect diagnosis, used mainly in late or complicated forms. In minor cases of salmonellosis, diagnosis is based primarily on stool culture, which is the gold standard test. Testing for Salmonella in suspected food items may be performed as a complementary measure, depending on the epidemiological context. Thank you for your attention. We remain at your disposal for any further information or questions.